We said one of the major jobs of our urinary system, our kidneys, is to be the excretory system. That is, it helps us control extracellular homeostasis by regulating the kinds of chemicals that we retain and excrete or get out of our bodies. This excretion process that the kidneys perform is made of three different processes. The first is filtration, the separation of the fluid component of our blood from the cellular component of our blood, followed by reabsorption. Much of what we filtered out in that fluid is actually chemicals we want to retain: water, glucose, amino acids, sodium, potassium. We want to retain most of that in the body. Even though it got filtered out, now we need this selective process to pull most of that material back into our blood. The third process is secretion. This is the selective process by which we can push certain chemicals out of our blood and in to the kidney tubule to become part of the urine and get out of the body. If our blood concentration levels of sodium or potassium get too high, we can secrete extra amounts of those into the urine to get it out of the body. Glomerular filtration was the first step to this excretion process. This is a passive process that relies on hydrostatic pressure, that is, the outward movement or outward force of blood against the wall of the capillaries. This forces materials through a filtration membrane that makes up the lining or walls of the glomerular capillary. This filtration membrane has three different portions to it. The first is the fenestrated capillary endothelium. Here, in our glomerulus, we have a fenestrated kind of capillary, which means that there are gaps or spaces between some of the cells. That gap or space provides an area for small particles to get pushed out of the blood and into the kidney tubules. But the fenestrations are small enough that cells cannot pass through them and stay trapped within the blood vessel. The second portion of the filtration membrane is called the basement membrane. This is connective tissue where lamina propria has woven together. It has even smaller gaps or spaces so that even proteins can be too big to pass through this basement membrane. We are filtering out even smaller particles like water and glucose and amino acids. Sodium and potassium are small enough that they can pass through this basement membrane. In addition to having these very, very small openings, the basement membrane also carries a negative electrical charge. Most proteins also carry a negative electrical charge. That means that even if proteins started to try to move their way through the small openings of the basement membrane, the similar electrical charge would repel those proteins and push them back into the blood. The third layer of our filtration membrane are what are called the foot processes of the podocytes, with these slit diaphragms that we find between them right here. These slit diaphragms are even smaller than the fenestrations that we saw before and do funnel material down through the basement membrane and through these foot processes so that only very, very small particles can pass through this kind of opening. Anything larger than a couple of amino acids would be too large to filter out of the blood in this way. We said that what's driving this glomerular filtration is largely hydrostatic pressure. Filtration at a glomerulus is much like the exchange we've seen at other capillaries. It involves a hydrostatic pressure, that is the outward force of blood pushing against the inner lining of the capillary. This is pushing material out of the filtration membrane. At the same time, because the blood is retaining the cells and large proteins, it's maintaining its colloid osmotic pressure that we had seen back in the cardiovascular system. This colloid osmotic pressure, caused by the retention of proteins and cells, provides an inward pull that kind of tries to pull material back into the blood. In addition, because we are pushing fluid out of the blood and into the Bowman's capsule, there is a certain amount of pressure that starts to build up within the Bowman's capsule. The Bowman's capsule exhibits its own hydrostatic pressure that pushes back against the outside surface of the capillary bed. Our net filtration pressure, that is, the measure of how much material gets pushed out of the blood at the glomerulus, can be calculated by taking the outward hydrostatic pressure and subtracting the combination of these inward pressures of the colloid osmotic and the Bowman's hydrostatic pressure. Our outward pressure in the capillary bed should be somewhere around 55 millimeters of mercury, whereas the inward pressure, the combination of colloid osmotic pressure and hydrostatic pressure of the Bowman's capsule, should only be about 45 millimeters of mercury. That means that our net filtration pressure that's trying to push material out of the blood at the glomerulus is only 10 millimeters of mercury. It's relatively small, but because the outward pressure should constantly be greater than the inward pressures, material should constantly be moving out of our blood and into the Bowman's capsule. Glomerular filtration rate, GFR, is the total volume of filtrate formed each minute. This is the combination of filtrate made by all of the nephrons in the kidneys. GFR is directly proportional to net filtration pressure. If net filtration pressure increases, glomerular filtration rates should also increase. GFR is also directly proportional to total surface area for filtration, as well as the filtration membrane permeability. If either surface area or permeability increases or decreases, it would cause a corresponding change in glomerular filtration rates. Even though our net filtration pressure is relatively small, only about 10 millimeters of mercury like we saw before, filtration is fast because of the large surface area that the glomerular capillaries have and the high permeability of the fenestrated capillary walls. This net filtration pressure is the number one thing that really controls glomerular filtration rates. This net filtration pressure can be controlled by adjusting the diameters of the afferent or efferent arterioles that are taking blood into or away from the glomerulus, respectively.